Christoph, you can hear us, right?
Yes, I can hear you.
Okay, cool. Yeah, so now you can see there is already quite a bunch of people gathered, about like 40 official participants and some more guests and less official and local students also. Yeah. So yeah, we are starting the third day of the school, and I'm very pleased to introduce Christoph Guger, who will be talking about BCIs and neuroimaging with ultra high density EEG. This is the development of their company called g.tec, and this company actually has been around for what I know from 2002, right, Christoph?
From 1999.
1999, yeah. Well, anyway, I know my colleague when he moved from the University of Southern California, when he moved to University of Washington, he bought back in 2001 or two, whatever, Christoph's system, and it was an amplifier, and it was a set of MATLAB tools that would allow, and Simulink tools, which would allow you to communicate with this device. And since then, the company is actually developing very cool solutions. And one very interesting fact about the company is not only making business, but making business, in my opinion, in a very smart way, especially a scientifically loaded business, because the company is organizing the largest BCI conference. And this year, the conference collected how many, like 70,000 listeners or participants? Or
So in the best lecture, we had about 88,000 people watching at the same time. So I guess if we count all the names, far beyond 100,000 people.
Yeah, so it's an online conference, and it's a huge coverage, and it's spread out over what, like 7 or 10 days conference, and yeah, and it's growing. Each year it's bigger and bigger, and it covers all various aspects of neuroscience, of neuroimaging, and of BCI. This is exactly what I mean when I say extending the notion of BCI. Okay, so now I'll shut up and pass the stage to Christoph. So Christoph, welcome.
Okay, thank you very much. So today I will show you some solutions that we have for ECoG and ultra high density EEG, like Alex already mentioned. So I'm sitting here in Schiedlberg in Austria. That's a very small village where we have 1,000 people. It's just a very good place to work because there's no traffic, parking slots, never ending, so it's good. In Graz, this is in the south where Arnold Schwarzenegger is coming from. We are designing our hardware. And then we have also offices in New York, in Spain, Canada, Hong Kong, Japan. And this year we started also in Cali in Colombia. We are running at the moment about 15 different research projects, and they all have to do with brain-computer interfacing. I just pick one example, seizure AI. In this case, in this project, we try to extract biomarkers that to detect epilepsy in a home monitoring environment by using deep learning. And here in B-Magic, for example, we work with nanoparticles together with ETH Zurich in Switzerland. And these nanoparticles can provide very precise electrical stimulation. And with magnetic fields around the head, we actually want to activate them to give very precise deep brain stimulation, for example, in Parkinson patients. But this is a project that will come to the market maybe in 10 or 20 years. So what I'm showing you today is how we use brain-computer interface systems for invasive and non-invasive brain stimulation and mapping. So here in the image, you can actually see a doctor in the operating room. He's having one of these ECoG grids in his hands. So the neurosurgeons are implanting the grids, and afterwards you can run neuromodulation experiments, and you can also stimulate the brain. And for these neuromodulation experiments, people are implanting up to 1,000 channels. So you can sense, for example, from prefrontal cortex to detect beta activity, beta oscillations. And this is, for example, a marker for tremor. And then you can on demand trigger an electrical stimulation in the thalamus. This region is called STN, and this is reducing tremor in Parkinson patients. But there are also fantastic studies in the area of depression and pain where you can do this. And this is actually Gerd Pfutscheler, my PhD supervisor at the University of Technology in Graz. And he came up about 50 years ago with the event-related desynchronization principle. And here you can see if a person imagines left-hand movement, we get an activation of the right hemisphere, and the red spot is called here event-related desynchronization, or shortly ERD. And if the person imagines a right-hand movement, then we find it contralateral on the left hemispheres. And this is very nice because we know exactly where we should place the electrodes to capture these hand movements. And this is a big advantage of EEG data, so we know where we should place it. But this is much more difficult for implants because you don't know where certain movements are decoded. And here is a slide from Kai Miller. He is a neurosurgeon at Mayo Clinic. Here you can see white and black dots. So every single dot is an ECoG electrode. They are made of platinum. And then you get cables coming out of the head and of the scalp. And they are connected to the brain-computer interface to run different experiments. So Kai was asking an epilepsy patient to be at rest. And then he calculated the power spectrum that you can see here in blue. You can also see here the characteristic 1/f damping in the power spectrum, so the signal gets smaller and smaller with higher frequencies. And then Kai was also asking the patient to perform a hand movement, calculated the power spectrum again that you can see here in red color. And if we look at the spatial distribution here, then you can actually see here the red area involved in the low frequency band. We can also compare the resting with the activity here. And you can see during resting, there's much more energy or power in the ECoG signal than doing hand movement. And this is called event-related desynchronization. I interrupt now quickly to let my dog in because she's complaining a lot. Just give me 10 seconds. So now she's silent. Sorry for that. So here you can also see the high frequency band. So this is between 80 and 100 hertz. And here it's exactly the opposite. Doing resting, we have less energy than doing the hand movement. This is called task-related high gamma activation. And if you look at the network, the cortical network involved for the high frequency band, you can see it's very precise. There's more or less only one electrode coding this information. For the low frequency band, we have a very wide cortical network involved. That's the reason why we can also detect it with EEG signals. And then Kai put a data graph on the patient and he was asking him to move 6 times the thumb that you can see here, 6 times. And then the patient was at rest and moved a couple of times again the finger. And then he used one single electrode, in this case, the dark blue electrode, which is located here on the ECoG grid, to reconstruct the movement. And it's indicated here in purple what's coming from the brain-computer interface. And you can see, we can even see the repetitions of finger movements. And very interesting is it here. So when the person is doing the first finger movement, we get a lot of high gamma activation. The purple signal is really big. Then it gets smaller with every single repetition, because you don't need the cortex anymore. The spinal cord is taking over. That's the reason why we have to do different activities to activate the cortex. You can also see for the index finger, we can nicely decode the number of repetitions. There's also this large overshot when the person is doing it the first time, then we don't have false positives. And it's even possible for the little finger. So here we have 7 repetitions and also the purple signal decodes it very nice. And this means the spatial resolution of ECoG is just very nice. That's the reason why we can do so many different things with ECoG signals. So we used it, for example, for our Cortex-U mapping system. And in this case, neurosurgeons are implanting grids like that. This is a 64-channel grid. You can see these small discs. They are made of platinum. And then we have very tiny wires going into the cable, and the cable is connected to the brain-computer interface. I can show you some videos of the past 20 years, what actually people did with all these activities. So this was the first experiment where Gerwin Schalk showed that an epilepsy patient can control a cursor with the brain-computer interface. So the goal is to hit the target, This was actually two-dimensional cursor control. This is about 20 years ago. Alex already mentioned our GUSBM system at this time. So it was actually the first brain-computer interface that could decode this ECoG activity and had also an API so that people could actually program new analysis, and Gerv Schalk put it into BCI 2000. And here is another experiment. In this case, Gerwin used BCI 2000. That's the toolbox he developed. On the left-hand side, you can actually see a 32-channel ECoG recording. And then he hooked it up to Doom. Some people might still remember it. So the patient used the joystick to control his avatar, and he was able to fire the gun with the brain-computer interface when the high gamma activation went above a certain threshold. And he showed it also to the US Army, and you can expect it. The US Army liked it a lot. Then this is an experiment that we also did like 20 years ago in Japan. And I show you the video so that you can understand how the setup looked like. So this was with Dr. Kamada and Dr. Kuni from University of Tokyo and Ashikawa Medical Universities. We are still working together with them. And in this case, we are reading in and Simulink the data with these blocks. So each block gives us 16 channels. And then we configure feature extraction and classification algorithms. And this is a typical recording setup. So these patients have the implant. You can see here the cables coming out of the head. They are connected to the biosignal amplifiers. And on the screen, they get certain instructions what they should imagine. For example, different hand gestures like a peace sign and so on. And on the right-hand side, you can see the recording computer actually running the experiment and also analyzing the data for BCI experiments. And then we tried it also in Japan in a telepresence setup. So in this case, we had an epilepsy patient in Ashikawa Medical University in Japan, and he controlled a human-like robotic body, which was far remote in another lab from Abdel Keda. And you can see he gave the commands with the ECoG system. And then the avatar did certain movements for him. So here the task was, for example, to get the Coke can lifted up. And then it was like a high level command. So with the BCI system, we made the decision and then the robotic system actually did it. This is also an interesting experiment. This is about 10 years ago. It's interesting because normally steady state visually evoked potential, so a flickering light can produce seizures. So people have to be especially concerned in epilepsy patients. For that reason, we developed a code-based visually evoked potential experiment. So instead of showing certain frequencies, we are embedding a certain code. So we have a certain on and off time of the flickering. And for that reason, not a stable frequency. And then we could actually run these code-based visually evoked potential experiments also with epilepsy patients. They achieved a very nice accuracy, so they could control actually the BCI system very nicely with this setup. And this is still one of the most important and powerful setups that you can actually do. And here's another experiment of a data set where we'll also show you a little bit later on an analysis. An epilepsy patient is sitting in front of the screen doing different hand gestures. And at the moment she is making a peace sign, a fist. All the fingers are straight. And on the recording computer you can see actually how the ECoG signals are read in, and the red bubbles indicate where the finger are actually located. This is our Cortex-Q high gamma mapping system, which can extract information after a few seconds. And I will explain it a little bit more in detail how this works. So for doing all these experiments, we developed a so-called embodiment station. And the idea is here that we have a patient where we put in an implant. And then we connect the implant to the biosignal amplifier, speaking with the real-time processing system. And then we can control in real time, for example, the auditory stimulation, haptic stimulation, electrical stimulation. So we can stimulate the whole body and we can observe what's happening in the brain. But we can also do it the other way around, so we can stimulate in the brain and we can observe what is moving of the body. And with this embodiment station, we can understand much more of the human brain. On the right-hand side, you can see an example where we put on vibro-tactile stimulators on all the 5 fingers. And then we just switched on for a few seconds the tactile stimulation. And at the same time, we are doing high gamma mapping, and this allows us to identify the sensory motor cortex for the middle finger here, for the ring finger, for the thumb, and for the index finger. And this is completed after a few minutes, so we can measure very nicely where activity is happening. This is actually a real-time video of a handshake. You can just see how quickly high gamma activation decodes hand movements. I play it again. So Kai Miller shakes the hand of the patient. And you can see we get the activation of the sensory motor cortex for the hand immediately. Just takes a few seconds. So this is very nice. And based on these results, Gerf Schalk developed the Siegfried technology. So here the patient is solving Rubik's Cube. On the bottom you can see a 64-channel ECoG recording. This screen gives the instruction to the patient to solve Rubik's Cube now. And here you can see the real-time high gamma mapping. So these red bubbles converge pretty quick. And then we can see the most important electrodes for finger movements. Afterwards, we record 15 seconds of baseline activity. And then the patient is sticking in and out the tongue. You can see the red bubbles converge pretty quick to a certain state. And after about 10-15 seconds, we have located the tongue region. Then the patient is stopping again so that we get additional baseline activation for the statistical analysis. The next task is to do a kissing movement. So she is now moving the lips. You can see the red bubbles converge again after a few seconds. And it makes also sense that the lips are very close to the tongue region in the motor cortex. The last task is to listen to a story. So all what the patient has to do is to listen to an English story. And of course you should understand what you are hearing so that we are able to identify the auditory cortex and Wernicke's area.
Mr. Jones had to go to New York. He decided to take a train. His wife drove him to the station, but on the way they had a flat tire. However, they arrived at the station just in time for him to catch the train.
You can see after these seconds, the short text, we have identified auditory cortex and Wernicke's area. This is a procedure that normally takes much longer. And based on this research, 2010, we developed our Cortex-U system. So this is a medical product. Also in Russia, it's a medical product that people can already use. And this is how it looks nowadays for neurologists and neurosurgeons. And there are very interesting facts because we can also extract temporal dynamics. So on the left-hand side, you can see the ECoG recording again. This is the screen giving the patient the instruction like relax and not to do anything for 10 seconds. And then we ask him to do certain tasks like stick in and out the tongue. This is very interesting because first of all, we get an activation here on the temporal base. This is the bottom of the brain because the patient can see the face, and this is just a face sensitive region. And afterwards, the tongue region gets activated because he was sticking in and out the tongue. Then the patient is relaxing again to collect additional high gamma baseline activation. Now he's listening. So that's an experiment that we conducted in Japan, and you can also see here the red bubbles because the patient could see the loudspeaker symbol, and this is actually the safe sensitive region of the brain. And afterwards we got auditory cortex and Wernicke's area. This is now Rubik's Cube. So first of all, we get an activation of this shape sensitive region and the color sensitive region on the temporal base, and afterwards we find the finger region over the sensory motor cortex. And this is super interesting because a couple of years ago we saw this here on the temporal base is an artifact. So nobody knew the meaning of these red bubbles, and nowadays, because of BCI technology, actually we can understand it. And Dr. Kamada, for example, does not resect these brain regions anymore, because if you cut out the face-sensitive region, then this has the consequence that the patient cannot recognize family members or other people anymore. This is, of course, something you want to avoid. And this here in the video is the golden standard. So this is actually what people are doing nowadays to do the brain mapping. So this is basically done in every hospital around the world. It's called electrical cortical stimulation. And in this case, an electrical stimulator is connected to the ECoG grids. And in this case, they are stimulating electrode 11 versus 16. And then they inject a current of 1 milliamp and they observe if she has any feelings, if there's anything moving, and then they write down, this is, for example, the middle finger or the index finger. And if they don't find a reaction, then they turn up the current to 2 milliamps, 4 milliamps, 8 milliamps, until they find a reaction, or maybe the region doesn't have any function. And then they go to the next pair of electrodes, like here, it's 11 versus 17. So this is the left thumb, the index finger, the middle finger, then they write it down. And basically they have to do every combination of ECoG electrodes. This is also the reason why this is pretty time consuming. So you can spend easily 1 or 2 days for doing all the mappings. And she has to report carefully what she is feeling or observing. And this makes it also difficult in children, for example, to do it, because they are not able to report it when they are very young. Now you can also see that sometimes they are on the edge of inducing a seizure, and then they have to lower the current again. Now she is reporting that this was the tongue region. And then he writes down electrode 12 versus 13 is tongue. In the background you can see the EEG technician connecting the ECoG electrodes to the stimulator, so they are doing it manually. This is now a picture naming to identify language area. So the patient has to say the names of the pictures that she's seeing. They try to interrupt Broca's area. This is the area responsible for forming words in your brain. When she's not able to do it anymore, they know they were hitting Broca's area, and now this is the most problematic thing. So they actually produce the seizure. This stops the mapping procedure, and it's of course exactly what you don't want to have, because it's painful. And the problem of electrical cortical stimulation, when we do it with cortiQ, we just sense from the brain and we don't produce seizures. And here's another application where we can actually map seizure propagation in real time. In the video you can see a patient sleeping. In the middle there's the ECoG recording of 64 channels, and on the right hand side you can see the high gamma filtered ECoG signal. And now you can see in the video that the patient slowly starts to move because a seizure starts to develop. So you can see in the high gamma scope the activity gets bigger and bigger, and on the left we map it with red bubbles. You can basically see that it starts in the middle of the brain, then it spreads out within a few seconds all over the brain. This is very useful information for the neurosurgeon, because he can try to interrupt the seizure pathways. And this is very nicely seeable with this high gamma mapping technique, which comes from the ECoG signal. This is what you actually need: a biosignal amplifier with 256 channels. You can also stack it if you need more channels. A computer for doing all the processing, and a screen which is giving instructions to the patients. Then we developed also different paradigms, for example with Dr. Ridaccio, a neurosurgeon or a neurologist from the US. And I already showed you a video, so people are relaxing, solving Rubik's Cube, sticking out the tongue, listening and kissing. And this gives us the most important centers of the brain. And then we have also a Dr. Kamada paradigm, where they also do Rubik's Cube, they read a story, they count mentally, They listen in this case to a Japanese story. This is the Japanese relaxing symbol. By having different paradigms, you can test different regions of the brain. This is, for example, a mapping from Yale University. This is how it looks, for example, for the finger region, for the kissing, the tongue movement, and the listening. This is printed out and then it goes into the operating room and people know what they should not cut out. We compared it, of course, also with fMRI. So here you can see the blue area, which is coming from the scanner, while people did a finger tapping. And then we looked also at the ECoG signal, and they are indicated with the red bubbles, and you can see they are perfectly overlaying. And here's an example of mapping receptive and expressive language areas. This image comes from picture naming. So this means we are showing a picture to the patient. So first of all, the patient sees the picture. This is activating the visual cortex here. So we get these red bubbles here first. And afterwards, they are forming the name of the picture in the mind. And this is activating Broca's area indicated here. Takes a few seconds and you can nicely find it. Receptive language is done by listening to a Japanese story. So first of all, you need the auditory cortex to hear what is on the loudspeaker, and then you need Wernicke's area to understand what you're hearing. And then you need Broca's area for formulating an answer to the question that was given to you. And also with this, we can study temporal dynamics very nicely. So first we get an activation of the auditory cortex, then from Wernicke's area, and afterwards from Broca's area. So very nice. And of course, it's also important to test if the results are stable. Here you can see we repeated the test 3 times. Basically, the red bubbles stay on the same position. This is super important that the results are consistent, because at the end, the neurosurgeon is removing brain tissue based on the mapping. And this is a comparison with electrical cortical stimulation. So in the middle in the brain, you can actually see the results that you get from ECS. This means they stimulate, for example, this electrode versus this one. And then the person is not able to name the picture anymore. So they needed a current of 8 milliamps. And then they know this is exactly Broca's area. And then they are stimulating here and here, and they get also picture naming. Blue is, for example, oral motor, so the lips and the tongue that you need for saying something. And white would be, for example, that there are no symptoms. And with ECS, you can also see that the visual cortex here is empty because it was not tested. With Cortex Q, we just get it as a side effect because the person is seeing the picture, we get the visual cortex activated and then Broca's area. So this takes here about 1 minute. This is 2 days of mapping. So we are much faster with that. And then there are also a lot of other fantastic things that you can do with ECoG. This is a study from Nuri Firat-Ince when he was at the University of Houston. At the moment he is at Mayo Clinic, and he worked with a Cortex grid, which is a combination of macro and micro grids. So you can see here these larger electrodes in blue. They have 1 centimeter spacing, and just in between they had smaller electrodes, micro electrodes. And then he put on a data glove onto a patient, and he instructed the patient to move a couple of times the fingers. And he used our g.tec Simulink environment for acquiring the ECoG data and also the hand movement data and also the high resolution camera images. And then he looked at the standard high frequency band between 60 and 280 Hertz. And you can see here at time 0, the patient actually moves the hand. So we get a little bit earlier already the brain activation because this is the planning of the movement. And then you can see the red spot goes all the way to 280 Hertz. So this is what most of the researchers are analyzing. And then he mapped with these green color boxes where the ECoG grid gets activated. You can see here, it's nicely distributed over the motor cortex. But then interestingly, he looked all the way up to 1 kilohertz. This is the ultra high gamma region. You can see that the brain gets activated all the way up to 1 kilohertz. And then he marked these electrodes in red. And you can see they have a different spatial resolution. It's even more precise. And this is very interesting because nobody knew before this study that high gamma activation goes all the way up to 1 kilohertz. And this is because traditionally this is hidden in the noise floor of biosignal amplifiers because the ultra high gamma activation is so small. And in our HIMP, we put in 4 signal processors to do a very high oversampling. So each ADC works with 614 kilohertz, and then we downsample or oversample to 2400 hertz. This produces a very high signal to noise ratio. And then we are also using 24-bit ADCs with a very high resolution. And we have also 2 co-processors doing a very high ultra steep anti-aliasing filter ring. And with all these techniques together, we are able to measure ultra high gamma, which is, of course, very interesting for many studies. And with this neuromodulation setup, we can also do very elegant studies. They are called cortical evoked potentials, or CSEPs. And there was Riki Matsumoto from Kyoto University who developed this technique doing his PhD. And here you, first of all, have to find, for example, Broca's area. And then you can stimulate only Broca's area, which is located here, indicated in red. So we provide a small electrical pulse exactly here. And then we look for evoked potentials at other brain regions. For example, here with the sensory motor cortex, here on the temporal lobe, where the auditory cortex is located. And this allows us to map cortical networks. So we stimulate here. And then we find a evoked potential, for example, on this electrode. And this is the motor mouth region that you actually need for giving an answer to a question that somebody is asking you. And at the same time, we find also evoked potentials here on the temporal lobe. And these electrodes indicate actually the auditory cortex and Wernicke's area, the areas that you need to understand words. So we can map the whole language network by just stimulating one pair of electrodes within a few seconds. And this is just a very nice technique for understanding cortical networks. And you can also probe many other cortical networks with these techniques, for example, pain or whatever you're interested in. You can do, of course, also sensory mapping. Here's a result of a patient where we did stimulation of the fingers. We used a high-resolution ECoG grid for this experiment. This means the electrodes are closer together so that we get a better spatial resolution. And you can see how nicely this is distributed. This is here the thumb, this is the index finger, the middle finger, the ring finger, the little finger. And we can get this result after a few minutes, 2 minutes of recording, and then we know exactly where the motor cortex for the fingers is located. Same is the case for sensory mapping. In this case, we do vibro-tactile stimulation, and then you can see the sensory cortex is just beside the motor cortex for the thumb movement, for the index, middle finger, ring finger, little finger. Takes also 2 minutes, and after 4 minutes of recording, we have a very good idea where these centers are located. That's another patient. So for every patient, this looks a little bit different. So this is, for example, the thumb, the index finger, the middle finger, ring finger, and little finger. So for every patient, you have to learn, first of all, where the electrodes are located. And this is something that we can also use for somatosensory evoked potentials. So in this case, we did a median nerve stimulation. That's a standard technique that's used in hospitals. So we put on an electrical stimulator and stimulated the median nerve here so that we got a nice finger movement. And then you can see how fast this mapping procedure actually is possible with this system at the moment. So I start the video and you can see that after a few stimuli, we already find these somatosensory evoked potentials. And here you can see the peak is going up. And on the neighbor electrode, you can see actually here that the peak is going down. This is called phase reversal, and it's actually the border between the motor cortex and the sensory cortex. And here we are also mapping these evoked potentials onto the ECoG grid. And here we can nicely see the border, the central sulcus between motor and sensory cortex. We put it also on MRI images, and you can see it follows the central sulcus precisely. And this is a very important landmark for doing BCI experiments, but also for neurosurgeons that they know where they are located inside the cortex. This is also an interesting experiment that we did with Dr. Kamada. So we put a high-resolution ECoG grid over the motor cortex, and then we asked the patient to make different hand gestures, like a fist, a peace sign, and all the fingers straight. And I show you now a video where the patient is actually doing the movement so that you can better understand what's going on. And this robotic hand here on the keyboard is actually controlled with the brain-computer interface, so with the movements that the patient is doing. And on the computer screen, you can see the instruction, what the patient is supposed to do. So at the moment the fingers are straight, you can see the robotic hand is stable. She makes a peace sign, the robotic hand copies the peace sign. Then the fingers are straight again. Peace sign, robotic hand copies the peace sign. It's very nice that we don't have false positives. We can extract the information faster than the robotic hand can actually move. So this looks all pretty nice, and this is also what we want to have to give a patient a robotic body back. So this was actually the goal of this VERE project where we developed the interface. But there's a very interesting segment in the video where she is making a mistake. You have to watch very careful. So the next one is a mistake. So watch careful. The instruction is peace. She makes a fist, and also the robotic hand makes the fist. This shows perfect embodiment. So the robotic device is actually doing exactly what she's doing, but it also shows that sometimes our subjects are making mistakes. That's the reason why you cannot always expect to get 100% accuracy of a brain-computer interface. That's another experiment that we did in this case with Dr. Ogawa in Japan. And in this case, the patient is just observing different images. And we are decoding with the brain-computer interface what he's seeing. So he's just observing. That's a face, and you can see we decode the face. That's a symbol. That's another face. A symbol. This is done by decoding information from the temporal base, where the symbol and face region is located. So we can decode faster than the patient realizes it. This is very interesting. He sees himself, it's a face. This is Christoph Guger from us, it's a face. This is also interesting. He's already used to Dr. Ogawa's face. That's the reason why he doesn't get immediately high gamma activation. It's the same story like with the data glove before. After a couple of repetitions, you don't get high gamma activation again. And very nice is also that we trained the brain-computer interface with artificial data on the computer screen, but we can recognize real faces with the setup. This is something that we also published in Frontiers for Young Minds. So this is a paper written for children between 12 and 16 years old. So it's very easy to read, but the nice thing is actually that it's reviewed by children. The comments that you get are very fascinating. And I move now a little bit quicker over this to our Pangolin system. So this is our 1024 channel high density EEG system. So we were fascinated by all the results that we get with ECoG signals. And then we were looking for ways for doing this non-invasively. This is inspired by this guy here, the Pangolin, because it has a lot of scales overlaying the body. And we wanted to develop something for the human brain. So if we look at this image, then we can see on top the skin and the scalp. And then we have some bones and the skull. We have the dura mater, the white skin covering the cortex, and then we reach the cortex. The distance from the skin to the cortex is between 15 and 25 millimeters. So it's actually quite a bit when we assume the electrode is sitting on the skin. And this is a typical 128 or 256 channel high density EEG system. You can see the distance between these electrodes is about 2 centimeters or even more. And this means the spatial resolution that we get is pretty limited. That's the reason why we developed these grids that you can see here. So we call the pangolin. One grid has 16 channels. They are made of gold. The diameter is just a few millimeters, and the distance, center to center distance, is 8.6 millimeters. So they are very close together, very precisely located. And then we have a connector in the middle to connect our biosignal amplifier head stage. So it's very important to have the amplifier as close as possible to the electrodes. You can also see we have to shave the head. We have to clean it with alcohol. Then we have little washers. They are filled with electrode gel. Then we can stick it on. We connect it to the head stage amplifier, and we can already get the 16-channel EEG recording. So here in the video, you can see how the recordings are planned. These are a montage creator. So we can take the pangolin grids or also ECoG grids from the library. Then we place it on the human subject. So altogether we can do 64 channels to overlay the whole scalp. Here you can see how we are filling the washers. This is with all the 64 head stages assembled. And this is a task where a person, for example, is doing handwriting. Then we can decode, for example, finger regions while they're doing it. And then we can map high gamma activation, also alpha and beta, onto the grids. And here you can see how the activation is changing during the experiment. And here you can see it with 64 channels mounted on this lady here. This is a comparison with the clinical standard. So the little rings that you can see here are coming from the pangolin system. So altogether 1024 channels. And the dark gray electrodes are the clinical standard 19 electrode system that they are using in hospitals. You can just see that one clinical electrode is overlaying 9 pangolin electrodes. So just the diameter is much smaller. And this also explains the better spatial resolution. And then together with the university in South Korea, we did a finger tapping experiment. So people were just tapping each finger a couple of times. We used the pangolin system to record the activity. And then we tried to identify single fingers. This is a task that's normally impossible with EEG data. But here with the pangolin system, you can actually see that the fingers are very nicely located. So the pinky finger, the small finger, is in blue. It's located here. In green, we have the ring finger. In yellow, we have the middle finger. In orange, we have the index finger. And the thumb is indicated here in black. And if we compare this now with the clinical standard, then you can see here with the pangolin system, we have this very nice distribution of single fingers. But if you would record only from here, from C3, then you can see you would be completely blind. So you would never get single finger activation with this recording. And with this spatial resolution, we can just do much more. And I am actually recommending to PhD students, select a couple of papers you like, replicate it with the pangolin system, and you will get much better spatial resolution, publish it again, and you're actually done. And in this paper, which was also published in Nature, we did central sulcus mapping that I showed you before with ECoG. So we did the median nerve stimulation. We had 5 subjects. We put them also into an MRI scanner to have a clear idea where the central sulcus is located. This is indicated here. So this is the boundary between the motor cortex and the sensory cortex. And here you can see the phase reversal. Here the black line is the central sulcus. And on the electrodes more anterior to this region, you can see the evoked potential goes up. This is the motor cortex. And on the electrodes which are posterior of the central sulcus, you can see the EP, the evoked potential goes down. That's the sensory cortex. And the border between the phase reversal is exactly where the central sulcus is located. And then we work together with Nuri Ince. They developed a very nice amplitude detector for evoked potentials, and then we plotted it again. Here you can see this central sulcus mapping with pangolin. It's just a very high and nice spatial resolution, and it perfectly matches to the anatomy that we got from the scanner. But it's completely non-invasive and also quick to do. And of course, you can also put this pangolin grids onto the arm to get hand gestures, but you can also put it on the chest to get a very precise ECG mapping, which could be useful, for example, to identify people with sudden cardiac arrest. And here's my last video, where we use the pangolin system for an artistic project with Anu Quipprecht. So she is a fashion designer, and she developed this fashion dress with a lot of actuators. And we hooked it up with the brain-computer interface, and then we could actually control all these actuators with the BCI system. And then my last slide is just an invitation for the annual BCI award. So the submission deadline is coming up again, 1st of September 2024. So we are running now the BCI award for longer than 10 years. And all what we would need is a 2-page description of your BCI project with a video showcasing what you're doing. And then we have an international jury looking at all the submissions. And then we select 12 nominees. And the nominees are invited to give a nice talk, and then the first place, second place, and third place winners are announced. And they get some cash, but they also get a certificate. They're also invited for a speech at our spring school in front of a large audience. And we are also invited by Springer every year to publish all the nominated projects in a book. The book was sold more than 300,000 times by Springer already. So at the beginning, we thought we will maybe sell 20 of them. So also the numbers here are very nice. You can also see a familiar face on the BCI award. Misha Lebedev is also here. So it's very nice to have him also inside this initiative. And with that, Alex, I would like to end my presentation. We reached the 45 minutes, pretty much exactly. And I'm here for some questions you might have.
Okay, thank you very much, Christoph. Let's thank Christoph for the talk. And now it's time for questions. Any questions? If you want to ask questions, please come here, because you need to talk to the microphone. And you can speak either Russian or English. If you speak Russian, I'll translate, but you come here. Microphones, these microphones aren't working today, okay? So, давай, по-русски, по-русски.
По-английски-то прочитаю или по-русски вам?
Ну да, да. Hold on, Christoph, please. Yeah.
ECoG-сетки, они записывают ECoG несколько дней подряд, и как они контролируют, что ECoG расположен на том же месте и...
Okay, yeah. I guess the question is about your ECoG-based mapping and how you control the location of the ECoG grid. When you do the replication study, how do you make sure that the grid stays at the same location? I guess it's more question to a doctor, but still, I mean, maybe you know the answer.
So the positions are dictated by the medical needs So if you work with an epilepsy patient, they go there where they want to do the epilepsy mapping. If you work with the tumor patients, they go close to the tumor so that the neurosurgeon can understand what he can cut out. And then these electrodes stay in for like 2 weeks. So it's depending on the hospital. Some people remove it after 1 week. And within these 1 or 2 weeks, we get the chance to do these BCI experiments. So ECoG grids are not designed as permanent implants because they have the cables coming out, but we can do these nice experiments in this time frame.
I see.
Sorry, actually, my question was about how do you control that the electrodes stay, well, the stability of their location? If
Okay.
for several days,
Yeah. Okay.
patients different.
Sorry, it was lost in translation.
That's a completely different question. So if you open the cortex, you have to open the dura mater, and then you can stick in the electrode grids under the dura mater. So there's a certain pressure of the dura mater to the grids, and also then they are adhesive, so they are sucked more or less to the cortex because it's a little bit wet. There's the cerebrospinal fluid, and they stick pretty nicely on the cortex, and normally they don't move. And yeah, so it's not really a problem that they move to another direction. They are sticked in.
Thank you.
Thank you. Any more questions? Yeah, go ahead. Then I will not translate. Okay, go ahead. По-русски, по-русски?
Да, давайте по-английски попробуем.
Ну давай, только сюда.
Hello, nice to meet you. Just for fun, what do you think? In the future, is it possible to run a Doom on the hippocampus or cell culture? Or cell culture?
This is something that people already did because you can record, brain activity from cell cultures, and then of course you can also hook it up to Doom. But this is without feedback. So this is just more or less random firing of the cell culture, controlling Doom. So this was more or less already done. Interesting is, of course, if you produce feedback. So if you see what's happening in Doom, and then the cell culture has to understand what's happening, and then you can control it on purpose. So that's a step that requires many more years of development.
Thank you.
Thank you. More questions? Yeah, come on here. If you have questions, please, could you please line up in a queue
Or сайт,
so that we don't waste
в
time.
чат напишите.
По-русски, по-русски.
Thanks a lot for your presentation, and I got a question about, is there research in the paradigm I read in one paper, participant asked to pull the trigger, and registration was from supplementary motor area. So there was a possibility to show the response earlier than participant got realization that he got even intention to pull the trigger.
This of course working, so we can extract with current BCI paradigms faster such comments than the person is realizing it. So I showed you the video with the faces and symbols, so where the person was looking at the faces and symbols, so we can decode faster than the person is realizing what he's seeing. And it's the same with finger movement. So if you are performing a finger movement, your brain gets actually active 1 second before, because there's a lot of processing involved. So you have to make up your mind that you move the finger, and then it has to flow all the way to the finger that you're actually doing it. And it's surprisingly long as 1 second. If you're already waiting for it, like you have a target in your gun or something, then you're already prepared, and this is reducing the reaction time, of course. But with the brain-computer interface, we are still faster.
One additional question. I actually was curious about what area of the brain processing this kind of mistakes? I mean, this causal relationship mistake, because you get a response earlier than you have conscious intention.
No, it's actually the preparation of movement. So all the preparations of actions in the body are decoded, of course, already beforehand in the brain, because somebody has to make the decision. If you're looking at error detection, then people are using mismatch negativity. So this peak appears about 100 to 150 milliseconds after you made a mistake. So this is when the brain realizes that this was a mistake. So if you pull the trigger, it takes you 100 to 150 milliseconds longer to produce mismatch negativity. You can measure it more or less all over the brain, and then you realize you made a mistake when you pulled the trigger.
Thanks.
Yeah, thank you. More questions? I have a question, or I have actually several, if you allow. We still have, what, 5 minutes left? So the first question, I guess, when you do the speech mapping, you primarily look at the high gamma activity, right, and the synchronization of gamma activity. When we are doing similar things, we observed over, like, I don't know, many patients, that we not only have the event-related synchronization of gamma, but we also have event-related desynchronization of gamma, and it's a very pronounced phenomenon. We see it like on every patient. I can at some point maybe send you pictures or whatever. So did you see this? And if you saw those, what do you think it could correspond to?
Normally it's an inhibition that you're also getting, so neurons get less active. And it also makes sense. So if the brain focuses on a certain task, then it's suppressing other tasks. So especially in pronounced in men, they are not able to do multitasking. So the alpha, of course, plays also an important role. So it more or less allows a cortical area to get active by reducing the alpha activity. And it makes also sense for higher gamma activity that the neurons get more silent while something else is done. And of course, the temporal dynamics is also interesting. So when certain actions are performed or inhibited, this is, of course, a very interesting field to study because there's not so much known about it.
Yeah, okay, thanks. And the other last question maybe, well, two last questions, but related to Pangolina. You show very nice results with Pangolina. I actually believe in high density mapping very much. But the question is, when you did the ERPs and showed this ERP result at the border between, the sensory and motor cortex, how many repetitions they were of, like, task repetitions?
So this done with median nerve stimulation, and you need maybe 60 stimuli. So it's 1 minute and you get it. So it's fast.
Not a lot. I see. Yeah. And also the last question, once you have received the ERPs, you show it all on the scalp, right? But did you try solving the inverse problem and just, do the classical thing to solve the inverse problem, look at the cortex, where it is, and etc.?
Yeah, we just submitted a paper with the source inverse problem solution, and then you find also the dipoles. So the mapping is very nicely and precise. And the big advantage of the Pangolin system is, of course, that they are precisely spaced. They have a much smaller diameter, so the source localization is more precise. With the standard 64 EEG channels, there's the liquid that you need. And then the diameter of the electrode is a little bit uncontrollable, and it's also very wide because a standard EEG electrode is already like 1 centimeter or more. And when you squeeze in the electrode shell, it gets more like 2 centimeters or even more because it flows around.
Yeah.
This makes the inverse problem solution much more difficult. Also, the electrodes are not precisely spaced. So sometimes it's 2 centimeters, sometimes it's 2.5. And this is all problematic. With the Pangolin, it's very precise because it's
very regular. Yeah. And the impedances that you get with each electrode, I mean each contact, what are the typical values for the impedance?
We don't even measure it, but it's much higher because the diameter is much smaller.
Yeah.
And we don't abrade the skin. But that's the reason why we have the active amplifier sitting on the electrode. So the amplifier must be as close as possible. Yeah. Otherwise, you would just record noise.
Sure. Yeah. Okay. Yeah. Thank you very much. If there are no more questions, I would like to thank you one more time. for the brilliant lecture and for the brilliant work that you are doing in developing the stuff and running this BCI conference and the BCI work plan. Yeah, thanks, Christoph.
Same for you. Very nice work you're doing.
Thank you.
Greetings. Bye-bye.
Yeah,
Enjoy.
bye. Thank you very much.